UNC-IMS, the Institute of Marine Sciences at UNC, now sit upon, as well as the Weapemiac, Lumbee, and Chowan Nations, who own the land surrounding the Chowan River, which is the study site of my ongoing research in northeastern North Carolina. So jumping into my presentation, just a little bit of background on me as a researcher. I actually grew up in the landlocked state of Kansas in the Midwest. So people often will ask me, how the heck did you become interested in marine biology? But the truth of the matter is my parents brought me to SeaWorld when I was a little kid, and I quickly became obsessed with everything about the ocean. So I was one of those little kids that decided I was going to be a whale trainer when I grew up, and no one was going to convince me differently. So I pursued a degree in marine science from the University of Miami. And during my undergraduate studies, I had this internship at the Mote Marine Lab, which was on the west coast of Florida. And while I was there, I learned about red tide blooms that were happening in the Gulf of Mexico. So this was the first research experience where I was exposed to the impacts that harmful algae can have on human health and also water quality. So when I graduated in 2019, I joined Hans's lab group at IMS as a PhD student in environmental sciences and engineering, which is technically a part of the public health school at UNC, because I wanted to study the impacts of these algal blooms on both air and water quality, and then in turn, human health and animal health. So jumping into the different backgrounds of my research, first is cyanobacteria or blue-green algae. So there's several different types of cyanobacteria that are found in all sorts of aquatic environments, but I primarily study those which are found in freshwater and estuarian waters. So we also refer to these as cyanohabs, the hab being harmful algal bloom or CHABs, and they're naturally occurring, but under nutrient enriched conditions, they can grow out of control and exceed the necessary biomass or amount of algae at the bottom of the food chain necessary to sustain the rest of the organisms in that ecosystem. So human activity is linked to the expansion of these cyanobacterial blooms due to that nutrient enrichment and also climate change, creating the perfect conditions for really warm, stagnant waters that are full of nutrients that allow these cyanobacteria to outcompete other types of algae that normally are also found at the bottom of the food chain. So the harmful part of harmful algal bloom comes from the production of these toxins, which are synthesized by several different types of cyanobacteria. But one commonly detected cyanotoxin in our North American waters is microcystin, which is shown here on the slide. So microcystin is a liver toxin. So it's linked in association to several liver diseases and cancers in human and animals. And it's produced across many types of different ecosystems, across many different types of cyanobacteria, but it's not consistent between seasons or between spaces. And so this makes it very challenging for us as researchers to forecast when people are maybe going to be exposed to these toxins. So we know quite a bit about the health outcomes associated with the ingestion of these toxins via contaminated drinking water or food, but we know much less about what happens when it's inhaled in the lungs. And this is all despite the fact that we do have evidence to suggest that the toxins and the cyanobacterial cells can be translated to the air via a process called aerosolization. And so they're found in aerosol. And some background on aerosol. In its simplest definition, an aerosol is a solid or liquid particle suspended in the air. And you might be familiar with other types of aerosols in your day-to-day life, such as cigarette smoke or sea spray. Also, aerosols are the reason that we wear masks to prevent the spread of COVID-19, as we create aerosols from our windpipe as we speak and laugh and sing, etc. So aerosols can stay suspended in the air for several hours at a time, depending on their size and concentration and chemical composition. The way that we name aerosols can get a little dicey, but in general, we also refer to them as particles or particulate matter, PM. And they are classified based on their size or their aerodynamic diameter. So this is one of the main factors that dictates their fate in the environment, as well as in the human lung. So the larger the aerosol, the more quickly it's going to settle out of the air and onto surfaces, whether that be somewhere on the ground or the inside of your lungs. And PM 2.5, or any particulate matter with a diameter smaller than 2.5 microns, stays suspended longer in the air and it can reach deeper into the human respiratory tract. So this is associated with more adverse outcomes. And this is all regardless of the chemical composition of the particle, which can complicate things even further. So chronic exposure to PM has been associated with lung diseases such as asthma and lung cancer. So in terms of looking at the concentration of aerosols in a unit volume of air, anything from 0 to 50 micrograms per cubic meter of air is considered good air quality. That's what you would envision like on a crisp mountain hike. But once you get over 200 micrograms per cubic meter, that's when you get to see air quality is dangerously low, and this can have significant health impacts in a community. So some of the most polluted cities in the world will see aerosol measurements up to 100 or 1,000 micrograms per cubic meter. So that's when you start to see this kind of image here that looks like a dense fog. So that's something we're interested in from the air quality public health perspective, is how many particles are there. And then also keeping note that the chemical composition can change the toxic impacts of the aerosol once it's in the body. Okay, so the mechanism that causes cyanobacterial cells and toxins to go airborne is a process called aerosolization, and it occurs at the sea-air interface here via bubble bursting. So what happens is wind and wave action entraps air bubbles beneath the water's surface, and as those air bubbles rise through the water column, all sorts of little particles and dissolved chemicals in the water can get absorbed to the sides of those air bubbles. And when they reach the surface and burst, they can create these aerosols that stay suspended in the air for several hours via two processes. The first being film drop formation. So these are when the little fragments from the top of these bubbles burst and shoot up into the air. And these aerosols are typically smaller and carry smaller, fine dissolved chemicals from the water. But there's also jet droplet formation, which happens when water rushes in to fill the cavity that air bubble once was there positioned, and then it propels this jet upward. And this fragments into little tiny particles that can result in larger aerosols that may carry larger particles from the water, such as cells. So this type of aerosolization is called primary aerosolization. And what I mean by that is all of these particles and liquids are emitted directly into the atmosphere in their liquid or solid form. But there's also some aerosols that are emitted initially as gases that later then condense in the atmosphere back into their solid or liquid form. And we'll touch slightly on that later, but mostly my research is interested in characterizing these primary aerosols that are produced via wind and wave action that might be carrying cyanobacterial cells and cyanobacterial toxins. So when I say my goal is to characterize these aerosols, what I mean by that is answer a couple of these questions. First, what shape are they? You can see aerosols come in all different sizes and shapes here, shown in these black diagrams. So this would be pollen here, this would be sea salts, but basically their drag in the air can be changed based on their shape. And so this can change their transport properties and how far they make it from the coast. Also, what is their average diameter? What size are they? Here in the bottom diagram, you can see there's all sorts of different particle sizes that are emitted, but mostly pollen, bacteria, plant debris are found in this coarse mode. And so these are larger particles that settle out sooner, that last for several hours as opposed to several days. But also keep in mind that some of these particles in these smaller modes, as they age in the atmosphere, they can bump into each other and collide and stick together, and that changes their overall size and number concentration. Two ways that we characterize aerosols is with their number concentration and their mass concentration. So these are pretty similar. You can see these boxes up here. The number concentration is different between these two boxes, but the mass concentration is the same. So this is just a way for us to keep track of how aerosols might change over time based on various weather conditions. Something like relative humidity can greatly change aerosol transport, because if you have a lot of water that's condensing on the outside of these aerosols, this can make them heavier and larger, so they settle out sooner. Or with increased sunlight, you see the opposite effect, where you would have evaporation off of aerosols. And then, of course, another thing we're interested in is what concentration of our target compound, the toxin microcystin, is present in each of these individual aerosols or in a unit of space. So all of this is to say that all of this information ultimately helps us forecast how far these aerosols are going to travel and thus the likelihood that they're going to reach human populations and be inhaled eventually. And then, of course, how severe their impact in the respiratory system might be based on where they would impact in your lung. So looking specifically at what we know about cyanobacteria and aerosols together, a handful of cyanobacterial species have been detected in the air of urban environments. So this means that we have seen aquatic cyanobacterial cells that can be transported several miles in aerosol and remained alive the entire time. But there really has been no clear association made between water toxin concentrations and aerosol concentrations. So this is likely due to differences between these studies in state of the bloom, the amount of toxin being produced, and also the weather conditions. But it's very difficult for us to just say, if there's this much toxin in the water, then it's going to translate to this much toxin in the air. But in the few studies that have quantified toxins in aerosol in both the lab and the field, researchers have found cyanotoxins along the magnitude of about 10 nanograms per cubic meter. And this seems low when you compare it to water concentrations, which is about 3 to 6 orders of magnitude different. But it's actually about the same that you would see for brevetoxin, which is a red tide toxin. So for anyone who's unfamiliar with the red tide bloom, which is the marine dinoflagellate that actually got me first interested in harmful algal blooms, brevetoxin is notorious for being aerosolized at the wave break on the beach and irritating the lungs of beachgoers. So this has been linked to asthmatic attacks and can really irritate people with chronic lung diseases like asthma or COPD. So although we might not be seeing as strong of responses to microcystin or other cyanotoxins being aerosolized at this concentration, we do know that it can elicit a response in humans for different toxins. So although there might not be immediate effects, we're still interested in trying to understand how much of the microcystin might be there in the environment, and also what the long-term effects on human health might be. There are many anecdotal reports of respiratory irritation following exposure to microcystin and aerosol, or people that are using water bodies that are experienced cyanohabs. So one research group found that recreational lake users were 2 times as likely to report respiratory irritation when using water that had cyanobacteria blooms, as opposed to those who were using water bodies that did not have cyanobacterial blooms. So we are seeing some types of irritation there. But to date, we really haven't had any toxicological studies that have really evaluated what exposure to cyanotoxins in this 10 nanogram range might do to human populations acutely or over time. So there's just a need to generate more information on the transport of cyanotoxins and aerosol so that we can make sure we're protecting populations using these bodies of water recreationally, but also just living near these water bodies especially. So since I spend a lot of time reading about this, my advisor and I recently published a review article on the aerosolization of cyanotoxins. So we reviewed papers in cyanotoxin chemistry in the environment and spray aerosolization and cyanobacterial studies in general, and also all the impacts that we know of cyanobacteria in the human lung. And there's one major unknown that I came across, which has kind of been the direction of my research for my dissertation. And that unknown is we don't know if toxins are aerosolized within the cells or as dissolved components of the water column. And I think this is a really important question to answer for the following reasons. First, going back to that red tide example, brevetoxin is so easily aerosolized because it's a hydrophobic compound, or a compound that has an affinity for air as opposed to water. And it's easily found in its dissolved form outside of the cell in the water. And this is because those red tide cells easily release their toxin at the wave break when they're broken up in all that rough wave action. And then those toxins are free in the water to interact with those air bubbles and then make it to the surface. So that's why brevetoxin, that red tide toxin, is so easily aerosolized. But cyanobacteria are really ecologically different. So cyanobacterial cells do not as easily release their toxins, and thus the toxin is much less likely to be found in that dissolved form, less likely to interact with those air bubbles and be aerosolized as fine aerosol. And then secondly, cells and dissolved toxins likely exist in aerosol with very different size distributions if you consider how large an intact cell would be compared to a dissolved toxin. So this would really change their size distributions and their chemical properties, and then also, of course, their fate in the environment and the human lung. So that would also impact three, the bioavailability between a cell and a dissolved toxin would be very different in the human respiratory tract. So they're going to impact in different places, the upper respiratory tract versus the lower lungs. And then also a cell versus a toxin would very differently be able to cross into the bloodstream. So by answering this question of whether or not toxins are aerosolized within the cells or as a dissolved compound, this will help us understand how likely they are to be impacting human populations, and ultimately what threat they might be posing to human health. So my research really is interdisciplinary in the fact that we have to look at the intersections of cyanobacterial ecology, water chemistry, meteorology, and of course, human behavior and what is going to get people more exposed to these toxins in the air. Okay, so jumping into my field research, where we're looking at this issue in the environment. In eastern North Carolina, the Chowan River in Albemarle Sound estuary has seen many years of back-to-back cyanobacterial blooms. And as early as the 1970s, the estuary had some pretty intense bloom issues. And this was tracked back to distinct point sources of nutrients that were being discharged into the river from agriculture and industry. But thanks in part to research conducted by my advisor, Hans Perl, at that time, the state was able to successfully reduce those nutrient inputs and mitigate the blooms for a while. But in recent years, the blooms have reemerged, and there's been several summers of blooms, chiefly composed of Anabina or Dolichospermum and microcystis, which are two types of cyanobacteria that have been known to produce microcystin, especially this microcystis. So if you look at this map here that was released by the North Carolina Department of Environmental Quality in 2019, it shows all of the different HABs that were reported across the state. And you can see that there's a heavy concentration of that cyanobacteria activity here. So during that summer, a microcystis bloom was reported, along with microcystin concentrations up to 620 micrograms per liter, which is alarmingly high. So this is more than 60 times the EPA's recommendations for swimming. So you definitely don't want to be touching or fishing in this water, really going near it at all. So due to this heavy cyanobacterial activity in this region, as well as the size of the fetch here, or the distance between the two shores, there's greater wave action at the mouth of the estuary. And so this is the perfect location for us to study the potential incorporation of cyanobacterial toxins into aerosol, because we see a lot of toxin being produced and also a lot of wave action. So we conducted our study in the summer of 2020, last summer, and the first aim was to survey the concentration of both microcystin and cyanobacterial DNA in spray aerosol. And we also collected water samples in parallel for comparison. And our second aim was to assess any environmental factors that might control the aerosolization of these toxins. So what weather conditions, what water quality parameters, what bloom factors might be promoting the specific aerosolization of these toxins and leading to a greater concentration of these cells and toxins in aerosol. So in order to carry out our objectives, we deployed these aerosol samplers at 2 field stations located on the Chowan River. Our campaign ran from June to September, which is the peak cyanobacterial bloom season. And in order to collect aerosols, we used these medium volume impactors, which are essentially giant vacuum pumps that pull air across a filter. And we use these cyclonic inlets here to size cut that 2.5 microns, since those particles are chiefly associated with more adverse health outcomes. And we also measured real-time aerosol concentrations with this little machine called a nephelometer, which was housed right under here. And so we use those real-time particle concentrations scaled with wind speed measurements and wind direction and all sorts of other meteorological conditions to try to estimate the relative spray aerosol production from wave action at both of our sites. And we also had water samples that were collected that we analyzed for cyanobacterial DNA and microcystin as well, as we did with our aerosol samples. And we also looked at all the other types of algae in the samples, as well as the cell counts, the cell density in the water, and also nutrient concentrations. So here are some more images from our work that we conducted. First, this is me changing a filter in a box that I engineered out of a upside down Tupperware to make sure that we were keeping our samples out of the wind and clean so that we could transfer these aerosol filters back to our lab for our analysis. Here you can see Dr. Karsten Baumann, which is one of our collaborators from the School of Public Health, who is helping us to deploy all of this aerosol equipment. So here he's seen showing us how to set up this meteorological station and also what all of the little trinkets on that did. Also seen as a fellow PhD student in environmental science and engineering, Naomi Chang, and one of our community scientists, Manuel Suarez, helping out there. Up here, you see another one of our community scientists, Kathy, who's helping us filter some of our water samples. And also just a fun little part of the summer, we went back every 2 weeks to switch out those aerosol filters. And there was a little family of ducks that would follow us around. So we got a time lapse of them growing up, which was kind of fun. But pictured here, some of our community scientists, I just want to give a huge shout out to the Chowan Edenton Environmental Group, who was that group of community scientists who worked with us in the Edenton region. So their organization strives to increase community involvement and environmental stewardship through hands-on research and teaching the local community about water quality in the Chowan River and surrounding inputs. And I mean it when I say that I could not have done any of the research that I did this summer without their help. So both of my field sites were actually located in their backyards. And each day that I was up in Edenton around 8 a.m., they were there with me to help lug equipment around and sample water and filter water. So I just want to give them all a very public thank you for the reports on bloom sightings and downloading my data at times and dealing with any time that I saw a spider in any of my equipment, and really just keeping me sane all summer. So I'm really, really looking forward to working with them again this upcoming summer. And I hope that they know that their knowledge has been invaluable to me. And I 10 out of 10 recommend working with community scientists if you ever get the opportunity. Okay, so moving into some of the concrete findings from our campaign. During our time there this summer, there was one large bloom event where we saw a surface scum, and this was at the end of June and in early July. So we captured some drone images here just for visualization of that surface scum. And this was chiefly composed of a cyanobacterial group called Anabina or Dolichospermum, which has been linked to microcystin production. And you can also see one of our collaborators from NC State, Ryan Pearl, here working to sample some of that surface scum. So this bloom event was associated with elevated concentration of cyanobacterial cells and also cyanobacterial pigment, and also chlorophyll A, which is a proxy for algal biomass. So we saw a spike in it there during that bloom. However, when we did see the very highest concentrations of cyanobacteria, we actually failed to detect any microcystin at all, which was surprising. But we did see microcystin produced at a couple different time points throughout the summer, including the second time point when we captured some of this large bloom. So this reiterates just really how challenging it is to forecast toxin production in the environment, especially when you have these big, dense surface scums that are very patchy. So although these blooms look really bad and they're not aesthetically pleasing, and the presence without toxin production can also cause a suite of environmental issues, this specific event was not associated with high concentrations of microcystin throughout our study. So even on the day that our drone imagery and then also the satellite imagery captured this dense surface scum, chlorophyll A was high, but our microcystin concentration was still less than 1. And this agrees with all of the North Carolina Department of Environmental Quality's monitoring in the system throughout the summer. So we wanted to look at if cyanobacteria cell density was associated with microcystin concentration in our data at all. And in short, the answer is no. The percentage of cyanobacteria based on our algal pigment analysis did not well explain the production of microcystin for this summer. So here you can just see some profiles of our chlorophyll A up next to our microcystin concentrations. And just a reminder that chlorophyll A is a proxy for algal biomass. But the maximum concentrations of microcystin that we measured this past summer were in the nanogram per liter range. So the highest we went was 30.6 nanograms per liter, which is well below 1 microgram per liter, which is the standard for safe drinking and swimming water. So you can see the spike in microcystin here, even though it was still low, was associated with that bloom that we showed. But we can't really explain why we measured microcystin later in the season at both sites in October. So this could have been a lag. Microcystin is very hardy and it sticks around in the environment. So it could have been produced several weeks earlier, and then we were just now measuring it at this time. But also, we're interested in getting our DNA results from these findings because there is a chance that maybe the types of cyanobacteria that were dominating at these two different time points when microcystin was being produced could be different. So it'll be interesting to see exactly what kind of cyanobacterial species were dominant and if they maintained higher concentrations of the genes that allow for the production of microcystin at these later time points. So getting a little more in depth with that microcystin data that we looked at, the approach that we use to quantify these toxins is called high performance liquid chromatography coupled with mass spectrometry. So this is a method that's extremely high resolution and it's very useful for quantifying low concentrations in our environmental samples, which is extremely important because meeting limits of detection for these toxins can be very challenging in aerosol. So although our microcystin concentrations were low in the water throughout the summer, the data that we did generate is form specific on that microcystin, and it reveals the entire chemical structure. And this was important for us to know because microcystin can vary at these two sites that are located in red circled here. So they have variable amino acid or protein side chains here. And those proteins, when they're different, can influence the toxin's chemical properties as well as its ability to become enriched in aerosol. So microcystins with hydrophobic side chains or side chains that have an affinity for air, such as microcystin leucine alanine and also leucine arginine or MCLA and MCLR are much more likely to be found in aerosol than their less hydrophobic counterparts. And if you look at our microcystin data, the two most abundant forms that we did measure were MCLR and MCLA. So these two forms could be easily aerosolized, and that's something that we are noting moving forward in our future studies. So moving into the results from this past summer, our initial scan of our aerosol samples did not reveal any detectable microcystin in our single day or our 2-week integrated samples. And this ultimately was not unexpected given the low concentration of microcystin measured in all of our water samples. So if it wasn't in the water to begin with, or it was very low in the water to begin with, we're not likely to reach limits of detection in aerosol or see a high concentration in aerosol. So although toxin production was low, there was still quite a bit of cyanobacterial activity, as you saw in some of our pictures and from some of our diagrams there. So we're still hopeful to see some significant DNA results in aerosol. So this work is being conducted with our collaborators at NC State in the lab of Ryan Pearl. And while I don't have any concrete findings to share with you just yet on this, I'll tell you a little bit about where we're at in the process. So we're specifically examining 3 characteristic regions of DNA to examine overall bacterial communities and cyanobacterial communities, and then also the genes that have the capacity to produce microcystin and aerosol. So those are the primers that we're using in our shotgun analysis and shotgun sequencing. So this is a work in progress, mainly because the filter that we impacted our aerosol on was a quartz fiber filter. And this was a compromise so that we could analyze that, we could split that filter up and analyze it, half of it for DNA and half of it for the toxin. And the extraction from it for yielding DNA has been a struggle, for lack of a better term. So after much troubleshooting with extraction volumes, the graduate student working on this was able to finally get a signal or a read from one aerosol sample. So we're moving forward with that method, and we hope to have some crude quantity data soon, before all of our water and air samples get sent off for sequencing. So we can see the amount and also the types of cyanobacteria that were in our water and aerosol samples. So as of right now, since we don't have a ton of aerosol data to discuss or analyze, what does that mean for my research? Well, we do have plenty of useful water data, including nutrient concentrations and algal community composition, and soon to be DNA specific information on the cyanobacteria there. So using linear regression or statistical approaches, we're going to be looking at how weather and ecological and water quality conditions might have impacted our DNA detection in aerosol. So for example, wind speed and direction can greatly influence wave action and spray aerosol formation. So these might be 2 factors that are strongly associated with concentrations of DNA in aerosol. Another example is how sunlight might influence the presence of cyanobacteria at the surface so that they would then be available to interact with those air bubbles and be translated to spray aerosol. So we're going to be taking a lot of little factors like this that all come together to see if there is a perfect storm of environmental conditions that might be good predictors about the aerosolization of this cyanobacteria. And I'm also using the historical data set provided to me by the NCDEQ to look at trends in the algal community structure and diversity over time, specifically cyanobacteria, the dominant cyanobacterial strain over time. So we're going to try to see if we can use the same linear regression approaches to understand why certain strains of cyanobacteria bloom in certain years as opposed to others. And this is especially important since strains like microcystis are associated with much higher concentrations of microcystin being produced. So if we can understand what factors are allowing the microcystis to be dominant in the system, this will hopefully allow us to better predict when microcystin is going to be present and when we should provide recommendations that people are not swimming or recreating near the water. So in terms of moving forward, what I've learned from the past summer, we've learned a lot about better meeting limits of detection of that cyanobacteria and aerosol. So meeting detection limits for any microbial life, especially aquatic organisms, and aerosol poses a challenge because it's much less concentrated in the air as opposed to the water where it originated. So next summer, we'll be using new pieces of equipment. The first being this biospot sampler, which is really cool. It allows us to sample aerosol directly into water or DNA preservative. So that allows us to concentrate our samples greatly so that we can better meet our limits of detection for our analysis. And also it's going to allow us to the sample so that then we can take the algae back to the lab, grow it up, and then run some further analysis on it there. And then we also are interested in using these high volume impactors. So this will just allow us to collect a greater amount of sample per unit of time. That will actually increase our sampling rate by about 5. So that would be really nice as well. And also with this equipment, we'll be able to use a different size cutoff for the type of aerosol that we're collecting. So what we had available to us in the equipment that was loaned to us by the EPA, we had to use a size cutoff of PM 2.5. And this might not be appropriate for sampling intact cyanobacterial cells in aerosol. So our goal is to use these cascade impactors, which are essentially multiple filters stacked on top of each other with a little space in between so that we can collect multiple sizes of aerosols at once and then differentiate them based on their size. And so our hypothesis there, since we think that cells are most likely aerosolized, since the toxin is not normally lysed into the environment, we think that we're going to have a higher concentration of cyanotoxin found in the larger aerosols or the intact cells that would become aerosolized. We're not going to be using quartz fiber filter again. And then also, since we're always constantly trying to battle the randomness that is chasing toxin producing blooms, we hope to capture more toxic conditions at some point in the next couple summers. Not that we're wishing toxin production at all, but we do hope to try to capture that in the environment. Sometimes it feels like when sampling cyanobacterial blooms, I feel like a storm chaser because we got to mobilize and follow the blooms whenever they exist. And then another exciting part of my dissertation research that's coming to light is that we intend to work on looking at the production of secondary organic aerosol from cyanobacterial bloom emissions. So going back to what I initially talked about, the difference between primary aerosol and secondary aerosol. Primary aerosol is the particles that are emitted as solids or liquids initially, whereas secondary aerosol is emitted as a gas initially, and then in the environment, it condenses into a solid or liquid. So plants, including algae, emit a suite of these biological volatile organic compounds, or VOCs. And so these are just gases that easily condense into their liquid form once they are in the atmosphere. And this is dependent on environmental chemistry and weather conditions, and also other pollutants that might be present in the atmosphere. And it's been demonstrated that algae from the surface ocean might emit some of these compounds, such as isoprene, which is extremely relevant in North Carolina, as this is emitted by pine trees. So there's evidence that these freshwater algae are emitting these VOCs that might turn into particles in the atmosphere. And as HABs expand and you see these really dense scums on the surface, it might be worth considering their huge contribution to secondary aerosol in the environment. So this summer, with some collaborators from the Public Health School who study secondary organic aerosol emissions, we'll be collecting water from the Chowan River and transporting these samples back to our labs so we can run some indoor chamber experiments to look and see if they are emitting these gases that can form these particles in the atmospheric conditions. So this is another way that water, aquatic algae might be impacting air quality. So that brings us to the end of my presentation for the most part. Just a couple more things. First, I just want to say thank you to everyone, all of my collaborators and lab mates and family and friends who have supported me to make this project possible, as well as my funding sources, especially NCC Grant and AppNep, who provided me the funds to conduct this research this past summer. I want to show everyone, not everyone, but a good amount of the people who worked on my team this summer. Ryan Pearl at NC State, one of his PhD students, Joel Sanchez, a handful of our community scientists, Manuel, Debbie, and Colleen, pictured here, who is the chair of the Chowan Edenton Environmental Group, Dr. Carson.
Uh-oh. Haley, are you with us? Check 1, 2. Well, it looks like we just lost Haley, everybody. We'll see if we can get her back into the show. In the meantime, well, at least we made it through the entire presentation. What incredible stuff. Go ahead, start dropping your questions and comments into the chat. And if we can get Haley back in, I know the team is working on it. Haley, are you with us?
Hello. How long was I lost?
Oh, we only lost you at, I think, the very end of the acknowledgments. And maybe just now again. Folks, let me tell you, we've been doing virtual programming here out of the museum. Now, let's see, today, the 1-year anniversary of our first virtual program will be Friday. And we have had an incredible run of providing great programming, nearly tech issue free. So this presentation is now just one of 2 or 3 where we've had problems. So I consider us very lucky. Hi, Haley, welcome back.
Hi, sorry about that.
No problem at all. I think we were going to go ahead and roll into, we'll see if we can play the video, if that'll work.
Yeah, let's do that. I'll play the video quickly. I'm not sure where I got cut off, but quickly, this video was created by a science communication intern, Abe Levin, who was able to work with me through my fellowship this summer. And I'm going to share a video that he made, a trailer for a documentary that he's created. So this is available on a website that we've made for that documentary, and we'll have a premiere sometime later this spring for the feature length film. Hold on, let me see if I can.
Generally, when people see bright green water, they know that that's not healthy. It looks like how toxic sludge would be depicted in a movie.
There has been a huge increase in the amount of nitrogen-fixing cyanobacteria in Albemarle Sound. Some of these toxin concentrations were really high, to the point where you definitely shouldn't swim in it. Way higher than the World Health Organization's guidelines for recreational use of the water. We don't really understand right now what's driving these blooms. Talking to some of the property owners up there, they're really worried about their own health. Being this close to these blooms, they're worried about, recreating in the water, or what kind of exposure they may have just from being out on their dock.
It's very important that the public and the community sees scientists as approachable individuals who really just want answers to problems that our communities are facing. When we go out and we do this research, we don't just want to collect data and then leave. We want it to actually have an impact on the community that we're working in.
All right, so that is the trailer, and that brings me to the end of my presentation. So I guess I'm ready for questions.
All right, thank you very much. Excellent stuff. And we do have some questions here in the chat in a few minutes to get to them. So I remind everybody, go ahead and drop questions. We're going to do that now. Okay, Glenn's got the first one. What happens to cyanobacteria that are inhaled deeply into the lower parts of the lungs? How long will they live there?
That is a great question. My inkling is that they're not going to survive very long viably. So you wouldn't see like a replication of cyanobacteria deep in your lungs. And we're not sure how they would impact. We don't think that they're going to cross into your bloodstream. So my indication is that they will be there for probably a couple days maximum until they degrade, and hopefully not have any impacts. There have been some studies that have collected, it's called a bronchial alveolar lavage, where they take liquid from the inside of your lungs, or not liquid, but just some of the mucus that's there, and look to see what's existing in there. And they have found cyanobacteria in there that exist in just the lungs of people going throughout their regular day, not even exposed to any sort of aquatic water body. So there is algae and microbes and all sorts of bacteria that are in aerosol that we inhale all the time. It's just a matter of whether or not that's toxic. And so far, we don't think that it's going to be a huge problem, that these aquatic algae would be living there, reproducing, wreaking havoc on your life.
Okay, well, that's better to know.
Yeah.
All right. Let's see. The next one here, Lisa writes, I love your story about wanting to be a whale trainer. What do you find the most rewarding about the work you do now?
That's a great question. I really love, I love teaching and doing outreach and talking to members of the public. I think that's one of the best things that I've experienced working with community scientists on this project. Is I like hearing feedback from people in the community, because as I kind of mentioned in the trailer, it's important that scientists are remembering why we're doing research. If our findings aren't going to be helping society in some way, then why are we doing it in the first place? So what I find most rewarding is getting to talk to people hands-on, just hearing their stories about how water quality has impacted their personal lives and what it means to them.
It's not doing the mass spectrometry?
That's kind of fun too, but.
Okay, there you go.
Yeah.
All right. Susanna writes, in North Carolina, is this issue unique to Albemarle Sound?
No, it is not. So we do see harmful algal blooms in other systems across North Carolina. So the cyanobacteria is especially bad in this region, but we also have it in places like Jordan Lake, down, I believe, in the White River, I think it's called, down near Wilmington. I might be misspeaking there, but you can check with DEQ to see all the different places that HABs have been reported, and anywhere where there is any sort of bubble bursting, you might be seeing the aerosolization of these algae. But the more wave action that you see, like in larger systems like Pamlico Sound or Albemarle Sound, the more spray aerosolization you would see.
All right, there you go. And a similar question from Carl. I realize your research is primarily marine, but can microcystin levels be elevated in freshwater bodies of water inland like lakes and streams?
Yes, so microcystin is actually primarily produced in freshwater. So the strains of cyanobacteria that produce microcystin normally start in freshwater, can be found in just lakes and little backyard ponds, but some of them can persist into the estuary environments. You're not going to see them as much in the ocean. So I'm actually very interested in what kind of microcystin production we're seeing across all the water bodies across North Carolina. So not just these big ones that are routinely monitored by the state or by the EPA, but also the little tiny backyard ponds and stuff. You also see cyanobacterial growth there. And we don't know how many toxins are being produced in those systems because we don't have the resources to monitor them frequently.
There you go. And I'm curious, and my question looks like will be the last one for today. But you mentioned early in the talk all of the different scientific disciplines that you're pulling in order to understand these algal blooms, cyanobacteria, and this toxin. Does it make it easier or more challenging to try to pull in, the weather and the way the water flows and the life history cycles of algae? Does that give you like a full figured picture of what's happening and make it easier to understand? Or is it challenging because there are just so many variables?
Yeah, I think you hit the nail right on the head in that it's really important that we're examining these complex processes from a lot of different angles, and that we're looking at everything that could be influencing these complex processes. But it definitely does make it more challenging to try to incorporate all of these factors into our statistical analysis, and also just I think that's where the randomness comes in. I think random is assigned to stuff that we don't understand. Like there's a reason that it's happening the way that it is, but there's just so many moving parts that it's difficult for us as researchers to understand which factors are dominating and how they're interacting with each other. So in short, I think it makes it more challenging, but it's necessary for research to be interdisciplinary like this in these environmental issues.
Well, excellent stuff. Haley, thank you so much.
Yeah, thank you so much for having me. It was a great time.
Excellent presentation. And I'll thank everybody for tuning in today to this presentation. Thanks for joining us. We will be back here again next Wednesday at noon right here on the museum's social media pages. So make sure you're subscribed to the museum's YouTube channel. Follow us on Facebook, Twitter, and Instagram. We're at Natural Sciences on those 3 platforms. And you can follow the North Carolina Office of Environmental Education. Their website is eenorthcarolina.org. And you can follow them on Twitter, where you'll get regular updates about nature happenings in North Carolina, including this lecture series. They're at North Carolina EE on Twitter. So I hope that I'll see you on the internet somewhere following the Office of Environmental Education or us, the museum. Lots of great programs happening all the time. Looks like we're going to be talking about sustainable oysters, or a topic thereabouts, next Wednesday at noon. So tune in for that one. Everybody, take care, stay safe, keep your community safe, and we'll see you again real soon. Bye, everybody.